Okay. Yeah. Thank you, everyone. Yeah, it's a great pleasure to have such a great guest, external guest. Yeah, that travel from Pretoria to Cape Town, and because I go quite a lot to Pretoria, it's quite a trek to come from Pretoria to Cape Town because you have an hour drive to OR Tambo. Yeah. And then you have a 2 hours flight, and then you have to come here and be here. So we really, really appreciate the trek that you did to come here today. Yeah, but especially because we are very excited about your topic. Yeah. So today, Professor Wanda Markotter, yeah, who directs the Center for Viral Zoonoses, yeah, will give us a presentation about how to understand bat-borne virus in South Africa. That's something that we are very excited, not only our group, yeah, but many groups in this building. Yeah, it's about the One Health approach. Yeah, and you know that Wanda is one of the leaders of this One Health approach, especially because I spent hundreds of hours with her as part of the NIP3, the National Institute for Pandemic Prevention application of the NRF, which was quite a circus, but was very exciting to learn about what all the groups were doing. Yeah, so it's a great pleasure to have external visitor, and it's a great pleasure to also see many of you. I can see that the direct email from Professor Nico to everyone highlighting, please. Yeah, it's very important because CERI, we invest a lot of money and time on these seminars because we find so important to expose our scientists to high-level science, but also to get new ideas to lead to collaboration. So it's a great pleasure, and we really appreciate all the people online. We have a big number of people online. We have close to 50 people online, and I'm sure that they're gonna join, and we have at least 50 of us. So at least your long trek here to the Tygerberg campus has not been in vain, and we really look forward for your presentation and today. And please save questions because one of the important things is to the engagement with questions. Thank you, Wanda.
Thank you, and can everybody hear me? That's the first thing. Online, please just tell me if there's any issues. So thank you for this invitation. It's really great to be here. I've only seen the building on press releases and photos, so it's really nice to see the biomedical building and also see a lot of people in person that I've been involved in discussions online for hours, like Tulio said. It's really nice to see everybody in person, and it's so great today to speak about my research again and not about high-level One Health policies for the quadripartite or other things. So I'm really going to focus on what I do in my research group in the Center for Viral Zoonoses. So I first just want to give you an idea of where this fits into the global stage, but also the broader thinking around pandemic preparedness, prevention, and response. And for this, I'm going to use one of our recent publications from the One Health high-level expert panel, where we put out an opinion piece of where prevention really fits into PPR and how important it is that we should invest also in prevention and not just in response and preparedness. And what we mean here is prevention of spillover of zoonotic diseases. So it's really that first case that spillover from an animal to humans need to be prevented. We're not talking here about stopping the disease in humans. It's really that first step. And this is really where my research focus on specifically this pinkish reddish color that you see on the one side, where we look at the host species that we consider potential reservoirs of some of these viruses in this case, where we look at the anthropogenic factors. We need to look at integrated surveillance, and we can talk for hours what we mean with integrated surveillance, and also looking at risk factors. And I'm going to try and show what we've done in this space, specifically with bat-borne viruses. So I have to say something about One Health, because if we talk about One Health, probably if I ask everyone in this room, what do you think One Health means, I'm going to get different answers. I don't think we always align what we mean with that. If it's a problem, I don't think it is. As long as we think about holistic approaches to address things and transdisciplinary approaches, the definitions to me is not that important. But the One Health high-level expert panel did put out a definition that's quite widely accepted in 2021, aligned with the quadripartite. And it says basically the same thing as the previous definitions, that we need to look at these interlinkages between environment, animal, and human health. But it also says that we need to bring in communities into these conversations. We still need to be multisectoral, we still need to be transdisciplinary, but it's broader than just, for instance, doing a project in academia. We really need to bring all the role players into this. And then really also addressing the issues while addressing the interlinkages between animal and human health. We will also address things like climate change, food security, water, clean air, because all of that also link to this interface of spillover of zoonotic diseases. So it's just a bit of a broad definition. And I think what's really also important if we function in this space going forward, even as academia, is the principles for doing that. Equity is extremely important. And this is across sectors, across people. There's so many different definitions of equity. Socio-political parity, and here we really mean that we need to listen also to the people that's affected by issues. And this is really your marginalized voices, your traditional knowledge, not just what we think is important from the scientific environment. And then transdisciplinarity is key to doing One Health approaches. It is a transdisciplinary platform and multi-sectoral collaboration. So in this space, where should we look for emerging diseases? So there's been some people that like modeling that brought out these papers on global hotspots. There's more than one of them. They sort of agree with each other of where the hotspots are. And this is done based on data that is available in the public domain, not very high resolution. So it's global data that we need to model. If it's also not there, we need to model what we think it tells us. So this is not context specific, also not geographically specific. So there's really this need to refine these hotspot maps based on data that we actually have for geographic regions. But meanwhile, we can use these maps to give us sort of an idea. I mean, it tells the same story where there's high biodiversity, tropical regions, you know, that's where we need to go and look for these diseases. But I don't think it's always that simple. But one thing that I wanted to show with this map, and you can't see it very clearly, but if you go down to the bottom, South Africa, there's actually yellow dots in South Africa. And this is in northern Limpopo and down our coastal region because we have high biodiversity and tropical regions in these areas. So traditionally in Africa, people always went to Central and Western Africa to go and look for filoviruses. And that's where most of our data comes from, because we were always worried about Ebola and Marburg only. But we also really need to look at some of these other areas in Southern Africa that's been really neglected. So just some background on that. What species? This is always a question. So again, using very, very broad data, we usually say bats and rodents, really important. We must also realize that bats and rodents are the two largest groups of mammals. There's lots of them. They all over, they're in different ecological niches. So it makes sense that they sort of come out on top, and we've sampled bats quite a bit. But they do account for that in these models, and they still come out on top. Some of the newer papers by Molenze and Stryker brings in some other mammal groups also, and actually say we shouldn't just focus on these two groups. We really should look at others, and particularly primates come up, avian groups, specifically due to avian flu. And then also, if you look at the pigs are actually not that far behind the pig group. But this is basically just the message is that we shouldn't just focus on one species. We should actually look at the whole ecosystem that can be involved in spillover of diseases. And it will also very much depend what pathogen you look at. Pathogens are associated with certain reservoir species. So it's complicated to design a biosurveillance program and really think of where you should focus, because you should know the literature quite well and you should know the gaps. It's not just storming in and sampling bats or doing surveillance. So getting to my program, so this is a program that started in 2011 when I was still working on rabies-related viruses in bats. And I now do very little rabies work. it's really grown into high consequence pathogen work, which is basically the phyllas, the coronas, the paramyxoviruses, and a little bit of influenza. And it's a typical academic program where we have postgraduate students, some support staff, some emerging researchers, some postdocs, but with a very strong international collaboration, as with most programs here also, internationally well funded by EU and also US Defence, but then also through the South African government, DSI, NRF, through my research chair, and through mechanisms like PRF. We do have some US collaborators, but also some collaborators in Europe. What is important about the program is that we decided a few years ago to focus on SADC. And the reason is, if you look at those global hotspot maps and some of the other data that I will show you today, Southern Africa has been really neglected in terms of biosurveillance in wildlife because we think the risk is not in Southern Africa. So it really, for us as South Africans living in Southern Africa, we said, let's regroup, move out of Western and Central Africa a little bit, and regroup and start doing work in Southern Africa and see what's going on and in our own country, South Africa. And that's how it started developing into this biosurveillance program, mostly focused on viral zoonoses around bat, livestock, human interfaces. So this is the site. I'm going to only tell you about one of our sites today, where most of our interesting work are happening. And this is a site in Limpopo that falls in one of those yellow dots on the global hotspot maps. And it's in a valley between the Wolkberg and the Drakensberg Mountains. It's where the Drakensberg Mountains actually meet the Wolkberg Mountains. And Tzaneen is on the top side, and then Polokwane on the one side and Burgersfort on the other side. So very rural area, but yeah, will this work? Yeah, so Tzaneen will be there. Let me just look at my computer, and then Polokwane there and Burgersfort on this side, about 50 kilometers. So it's really in the middle of this area. Yeah. It's that one there at the top. There. Yeah, right, sorry. I must use my pointer. And what's nice about this site is it's really this interaction of this rural housing. You've got the agricultural intensification and places where people do agriculture. You've got livestock roaming. There's certain areas that's controlled areas for livestock feeding, but then they're also roaming like in all other rural areas just all over. And then why we're interested in this specific area is because of this cave called Matlapitsi, where we have a maternity roost of Rosetta aegyptiacus, or the Egyptian Rosetta's fruit bat. We think population size estimates are always difficult, but it goes between 6,000 to 20,000 bats at certain times of the year. And they then also have their young here, and there's reasons why this is important. It's also mixed with about 9 other bat species that also inhabit this cave, including the horseshoe bats that's very much linked to SARS-related bat coronaviruses. So really a nice interface between fruit-eating bats, insectivorous, and livestock and people, and everything happening in this one environment. So we've been working here since 2011. So it's really been a long journey working in this area. Why are we so interested in the Egyptian Rosetta bat? So this bat occurs quite widely spread all over Africa. It's probably most well known for being the Marburg virus reservoir, and there's very good evidence of that. We know this is one of the reservoirs for Marburg virus. It's a cave-dwelling fruit bat, also the only cave-dwelling fruit bat. The rest of the fruit bats live in trees and not in caves. So that also makes it quite a unique situation. And it's been associated, I always say, if you test Rosettas for anything, you will find it. There's always some virus in it. It's just associated with almost all the virus groups, including influenza and the ones that I will talk about today. So one of the things that's really nice about the work that we do, and we're able to do that, I think, because we're in country, is that we can do longitudinal surveys. So we can actually go to our sites every month and get seasonal data over time, over years. It's not so easy if international groups come in to do that, because you can maybe come in for one season and go out. but we're really in a situation where we can do this. And we then sample several excretion samples for viral testing, which is your fecal, your urine, saliva, and then also blood for serology. So we completely moved away from some of the work that we've done historically when I started in this field, where we were taking voucher specimens and testing organs, to testing non-destructive samples and taking excretions from bats. And it's been quite successful. So it just shows it can be done that way. You don't need to test a liver of a bat to find a filovirus. And then we also broader to this, we collect all morphological data. So it's measurements, it's sex, age, all of that, together with environmental data and land use data that we can use. Something we started with quite a few years ago that makes the interpretation of results interesting is marking the bats. And we moved to a tattoo system. It's actually just a human tattoo system that we tattooing bats with. So that if we recapture bats, we actually know what the bat was. We also started pit tagging, where we can actually then use antennas to see if we can do population estimates. And then we do lots of other tracking methods also that I will talk about. So this is sort of, it's one of my postdocs pictures, but it's a surveillance blueprint. And we always say, you know, we're looking at pieces of the puzzle, but in the end, all the pieces needs to come together to really have answers about risk. So yes, you can sample the bats. You can go to a cave and you can sample bats and you can test them for viruses. You will get a high diversity. You can characterize those viruses, but it can't stop there. That just tells you what's there. Then you need to go a bit further. You need to overlay this with other species in the area that may be of high risk. Use maybe things like serology to look at spillover. Look at any human activity that can increase contact. Because if you just have the viruses in bats and there's no contact interface, nothing will spill over. It's really about that contact interfaces. And most of the studies that's been done don't really address that and look at that in detail. So really putting all these pieces of a puzzle together is really important, and this takes time. It's not something that you can start tomorrow with all these aspects. It is sort of a staggered process that you can add as you do research over time. So getting to something that you probably know quite well is coronaviruses. So I'm not going to tell you too much about the structures and all of that. I'm just going to highlight a few things in terms of bat coronaviruses. And this is a part of my program that's driven by Marika Geldenhuys-Venter, who is one of my emerging researchers. And really looking at bat diversity coronaviruses in South Africa and trying to understand the risk of spillover. And what's important is just the 4 emerging viruses that we saw in the past few decades. SARS coronavirus, SARS coronavirus 2, MERS, and then one that only spilled over to pigs. We never saw it in humans, but it's something we should watch, and that's SARS coronavirus. Now, all of these coronaviruses have some link to a related bat virus, and the word related is important. Not exactly the same virus. There must have been some evolution, some spillover somewhere where these viruses adapted to, in the end, become human outbreak viruses. What is quite clear with coronaviruses, there's a huge diversity. If you go and collect fecal tomorrow from bats and you test it for coronaviruses, you will find a coronavirus, alpha, beta, all over the genera. It's just this huge diversity. It's quite interesting. They do cluster according to bat species. So you would have viruses that's just specific for Egyptian fruit bats. You will have one that's for horseshoe bats. So they seem to be really species specific with just a little bit of sharing here and there. So high diversity. And I'm gonna, so in Africa, we summarized the amount of studies that's been done, and you can see on this map, so the color shows if there's more than one publication. Most countries just have these once-off publications, publish it. Yes, we have a diversity of coronaviruses based on a very short nucleic acid sequence, and that's where it stops. And we can't really make any inferences about spillover risk from that. We just know there's coronaviruses, and we know that actually already. So it's just adding more data to that diversity. But what we do know about African bats, and it's associated with very specific species, is that we have 229E and NL63 related viruses. In bats, we have SARS-related viruses in horseshoe bats. Very limited. It's only a study in Rwanda, and then we actually also identified in South Africa, but very far from SARS coronaviruses in humans. Very, very different genetics, not the same virus. And then we have MERS-related viruses that we've also identified in Africa bats, but also in South Africa. Wolfgang was one of the people that actually published on that, and then my group also. Yes, yeah. Very, probably very long historic history of bat viruses with those. It's not a spillover that happened yesterday. Yeah. So our study methods, I'm not going to go into a lot of detail. Coronaviruses, interestingly, in humans, you see a respiratory disease. In bats, it's a gastrointestinal tropism. So the viruses are in the fecal material. And that's why we target fecal in biosurveillance studies for coronaviruses. And you can do it in different ways. You can put sheets under populations of bats and just get pools of fecal. You can't say from what bat it came. You can't say sex or anything else. But you get good population data. Or you need to catch the bat and you actually need to give it a rectal swab. And then you have some additional data that you can use. So we do both. And we then overlay this data. We then also try and do genomics, and I will say a little bit about that when I get to one of the slides. So this is just some of our Egyptian fruit bat data. I mean, if you start putting other bats in here, these trees look even more crazy. It's just all over all kinds of coronaviruses based on short sequences. But I want to highlight specifically the work that we've done on HKU9 in Rosetta's bats and a few interesting things that we're starting to see because we're doing longitudinal surveillance. So we do find novel viruses. That blue block that you're seeing there is a novel alpha coronavirus, then another beta coronavirus that also new, but what does it really mean? You know, we're just seeing it, we can characterize it with full genomes and say we've got a new coronavirus, but that's all we know about it. But HKU9 is a really interesting virus, this red that you're seeing here, high prevalence. So we're going over 10% in bat populations over time. And then I'll highlight a few other things also. So why? This is an Rebecca virus. It's not part of the Rebecca group, but it's an Rebecca virus that's been identified in several fruit bats in Asia and in Africa, always in a high prevalence in populations. So there seems to be high excretion peaks and in a high diversity of fruit bats. So everybody is sort of looking at this virus, you know, it's usually a warning sign if it's several species that can be involved and if you've got high prevalence. And some of the work that we then did, in addition to that tree that I showed you at the beginning, that is just a very short region of an extremely conserved region of the polymerase gene, the viruses look very similar if you look at that region, but then start looking at spike proteins. So in this case, we actually have more data now, but we did 41 of the spike genes. And this is a virus that circulates in one population of bats at certain times, and we're getting 8% differences in amino acids in the spike protein. So that already tells you there's a lot of mutations going on in the specific population with this virus. But we could also, so we identified 5 groups, and what we're doing now over time is also looking if some of these variants outcompete each other or are dominant in these populations, because we've got this data over 3 years. This is 3 years data. We actually now have another 3 years that we can add. So we're really looking over time, and we're seeing interesting population and maintenance of viral dynamics where some of these variants are outcompeted at some point and they disappear. They come back probably when there's no immunity anymore. So how this exactly happens in bat populations, we still need to look at. What's even more interesting is if you really go to the receptor binding region and you look at the amino acids of these spike genes that we sequenced, you're going up to 16% difference, 1/6. So there's high diversity in this one population in terms of the receptor binding region, which means these viruses probably have the opportunity to adapt at some point to spill over to other species. So just some things that you can infer from genetic characterization beyond doing very small regions of the polymerase gene and just saying you find a virus or a piece of a virus. We're also doing genome characterization, and this is after I've showed you spike gene information, this is really to look at species demarcation of coronaviruses, because we need these other pieces of the genome to really say this is a new viral species. So, and we could, with these three viruses that I'm showing to you, when we have full genome sequences, we can actually show that. Now, I think most of you are probably in this area of sequencing genomes of human viruses after you've seen disease. With this type of work, it's extremely tricky to do full genomes. So there's this drive worldwide where people say we should do full genomics for biosurveillance also, just target genomes, just do NGS. It is really difficult. And the one reason is the bats are not getting sick. So low titer viruses, very low titers. The other problem is the diversity of these viruses. You can't map them to reference genomes. So just mining it out of all the noise that's coming with your bioinformatics is extremely difficult. There's now fancier ways of doing it, but it's expensive. So we're really struggling to get full genomes for bat viruses. It's really not an easy thing to do, but it is necessary. And we can't just stop with publishing short sequences all the time, because we need this genomic information to also make some conclusions. So overlapping all of this now with the population ecology became very important. And because we're sampling over time, every month, we can now look at the reproductive seasons of the bats in this specific colony. And we know with Rosettas that the bats come, a large size of the population come to this cave in spring, because we then see a spike in the population. They then mate. They have their young around October, November, and those young become independent, which means they're not going to fly with their mothers anymore, around February to April. And if we look at our virus peaks, this is just all the coronaviruses together, and this is a GLM and a GAM model with the fecal rectal swabs and the fecal from population collected. We're seeing these peaks probably when we're seeing a waning of maternal antibodies. We're not sure how long duration of maternal antibodies are in sub-adults, but the peaks are in some way driven by the amount of sub-adults in the colony. We're not exactly sure if it's a reproductive trait or if it's really the sub-adults driving it. We're busy looking at that in more detail. But you definitely have these seasonal peaks with coronaviruses, and the rest of the year it's almost gone. And then you have a peak again that's driven by the reproductive seasons of the bats. And this becomes important if you talk about risk and risk periods for spillover, because this is the times when we have sometimes up to 40–50% not seroprevalence, nucleic acid prevalence in the populations. So we know with all the coronaviruses, we see the seasonal excretion linked to reproductive biology. We think age is important, and that we mean sub-adults are probably playing a role, and waning of maternal antibodies. Sex, we've never found to play an important role, if it's a female or a male. One of the other things that I mentioned at the beginning is recaptures and infection rates. So because we're tattooing the bats, we recapture some of them. Now, it's not so easy to recapture them. You're sitting with a 20,000 population of bats. We can catch about 100 a month, so you don't get a lot of recaptures. So the few that we did get is giving us some interesting results in terms of catching them now, and then when we recatch them, are they still positive for a coronavirus? And we could see with the coronaviruses that it seems in 4 weeks, they become negative. So we're looking at about a 4-week clearance of infection with some of the recaptures. And this can become important if we're looking at models of maintenance of viruses in specific populations with actual data. And we also see that it's actually obvious alpha and beta coronaviruses, there's no cross protection because the one will be infected with the alpha the one day and with the beta another day. So this is some interesting data that we're building on over time. So getting to so what? We do all this work in bats, but what does it mean and how can we actually say what of this is important for emergence? And this is extremely difficult. And just to show a few examples, I think we all know these examples. So when we saw the SARS coronavirus outbreak in 2003, we started doing surveillance for bat coronaviruses. We found some closely related ones. We never knew that's where we were supposed to look. We found some closely related ones through the years, some more closely related ones. And in that group of viruses that we actually found, there were ones that were related to COVID and to SARS coronavirus too, but we never flagged them because they were not related to SARS coronavirus, the first one. So very difficult to say which ones are going to cause the next pandemic. The same with MERS. We knew years before MERS started becoming established in camels that there's related viruses in bats. We never said, oh, this is going to cause an outbreak. So it's not like we don't always have this data. I don't think we always know what to do with the data and how to flag which one will be important going forward. So there's ways of doing this, and it's all to do with modeling and mathematical models and assessment tools. And one of them is the spillover ranking tool that's been published a while ago. And this is basically ranking viruses. There's a website. You can actually go and type your virus in there, and then you see where it ranks. But it's trying to bring viral characteristic, ecological characteristic, geographical, human behavior, and all of that together to rank some of these viruses in terms of spillover risk. And HKU9 has been ranked as number 16, the one that I showed you that we're finding in Rosetta's in South Africa, because of the data that we have, that it's very diverse, there's mutations, high prevalence, high species diversity that it occurs in. So this is some of the tools that we can use, but I think what is really also important is that we shouldn't just stop with surveillance. We should start looking at things like virus culture. What receptors can these viruses recognize in a biological system? Animal models, which is really extremely complicated to do, but there is a need to still do that. And we actually in South Africa have those opportunities with the lab at NICD, the Special Pathogens Unit, where we used to host bat colonies and really could look at some of these questions. So at the moment in our study, we're just really understanding the diversity. We're understanding a little bit about the virus characteristics. We can look at the spike protein. We can look at receptor binding units. We can try and rank based on this, how important it may be in spillover. But it's really a very small part of the puzzle. So one of the others, before I get into the last part of the talk, just highlighting one of the other viruses that we also do and how different it can be between virus families. It's not always even the same between viral families that we consider as potential zoonotic viruses. It's the work that we've done in the paramyxoviruses, and this is work done by one of my other emerging researchers, Miranda Mortlock de Vries, who's really driving this part of the program. And here I'm only going to focus on the Nipah viruses group, and this group is associated with fruit bats. You may know the Nipah virus group as Nipah and Hendra virus. We don't have Nipah virus in Africa. We've got related viruses that we've identified that you can see here. They're called Ghana Nipah and Cedar virus. They've got all kinds of different names, but they occur on the African continent. Nipah mostly associated with Bangladesh and Malaysia. We know it spills over. We know it causes human disease. People have died from that virus before. And Hendra associated with horses as an intermediate in Australia and can also cause human death. So we started doing surveillance in South Africa also for these viruses, very much the same as with coronaviruses. But another sample type that's really important with henipaviruses is urine. That is one of your best sample types for the tropism of henipaviruses. Now you can think getting urine from bats is not so easy. It's not like they're giving it to you on demand, or it's really tricky to get it in the hand. So most of the time you try and do sheets also under roosts. And then you run the risk of having fecal and urine contamination. So there's some trade-offs of doing that. But we tested hundreds of samples, no surprise, found henipa-related viruses in Rosetta's fruit bats. And this is just what you can see here in red. So there's quite a high diversity. And when we started looking at this, and this was still some of the work done by Prof Drosten lab, where he put criteria out, even on short sequences, how you can say if it's putative viral species. So if you use that criteria, we came up with 18 different putative henipaviruses in this, henipa-related viruses in this colony. So this is just what you see here. And it's also, the tropism is very different. And then even if we go down and we look at what sample type we detected it, between these three shown at the bottom, some of them will be more in urine, some of them will be more in fecal. If you look at seasonality, they even have different seasonality profiles. So if you look at these things holistically, just as paramyxoviruses, you're missing these things. It's very, very specific to specific variants or specific species of viruses within even a specific virus genera or family. So just to show that it can be complicated, and then if we look at the excretion dynamics, same if we look at the reproductive season of Rosetta's bats, the excretion dynamics of paramyxes are at a different time point in the year. We're seeing it June, July, where we saw it earlier with coronaviruses, and we're seeing a second peak of paramyxes in September. And we think that may be something to do with when the bats are giving birth, or stress, or the recolonization of more bats coming in. So a completely different picture than with the coronaviruses if you look at time points in the year, just to show how complicated it can be. So as I said, we're building the first part of the picture, but what's next? So this is not where it can stop if you really want to look at prevention, prediction, risk mitigation. You need to start looking at serology, and I will say why we didn't start with serology, because it sometimes doesn't make sense. Why not start with that? You need to look at contact interfaces. You need to look at surveillance among other terrestrial animals and also humans. And then understanding human behavior is really important. So the last part of my talk is really moving into this. And I'm a virologist, so this is completely outside of my expertise. But it was essential in the program to bring this expertise in to start answering some of the questions. And really building a really interdisciplinary group. So we started looking at movement patterns, just doing some simple things originally. And this was some of the work of my postdoc, Lau de Vries, where we look at fruit abundance, because Rosetta's Egyptiacus are known to eat figs. That's what they like the most. So looking at wild figs versus figs in somebody's yard, or looking at when there's no figs and they may be going for the chief's mango tree, became important. We had to map fruit trees in the area to look at contact interfaces. So we started doing some vegetation and vegetation mapping work. Over season, we started looking at contact of humans, livestock, and wildlife. We looked at the diversity of wildlife in this area. And I'll just using camera traps, and then also looking at other bats sharing this space with Rosetta. So this is just some of the camera trapping that we've done. We've put camera traps at caves, and we would see what animals and people are entering the caves. And there's civets entering, there's goats, there's people, there's dogs, there's monkeys, there's porcupines, giving us an idea of what interface we're dealing with. We can do this better. This was just a start, but it was really just to see what we're dealing with in this area. One of the papers that was actually published last week by one of my MSc students, Matthew, we put radio trackers on the fruit bats. And the problem with tracking with fruit bats, and specifically this fruit bat, there's no trackers small enough for their body weight so that you can get long-term data, not GPS or anything else. So you can really just look at short-term data. The one problem is also they roost in caves, so you can use none of the solar batteries, that's light. So they can't charge while hanging in a tree. So you really get very, very limited data, which basically just give you foraging data. You can't see if they're migrating somewhere else. The data is just not that advanced. But what we did here, and this was just radio tracking, student running around with an antenna for 2 years in this area, trying to see where the bats are every evening. The red is where the housing is. This is in the summer months, this is in the winter months. And although very basic data, it tells us something really important, that in the summer months, they're in the valley where there's no houses. This is where all the wild pigs are, because they have enough food there. But in the winter months, they start moving into the dwellings. And we could identify bats in specific fruit trees in a little dwelling where they have that contact interface. We then followed this up with GPS trackers recently with my US collaborators. And here we could get 4 nights of data. And very interesting, if you look at this, there's some bats that they fly 300 kilometers that evening and go and sit in a tree in a random place for some reason, and then they come back the next day. We're not sure why they do that, but they do. And then, it's got a girlfriend or something somewhere. And then you have most of the bats went into the valley again. But it's interesting if you look at this, there's those 3 dots are in between houses, and it's actually associated with specific fruit trees. And then that is a meeting place. So in these rural areas, some of you may know that usually if there's a big tree, that's where everybody meets. So this is the community meeting place. So that's why it was important for us to show that dot, because there's interaction with people. And it's also where all the dipping tanks are for all the animals. And then this map is where they're flying over the mountain to Zanin and go and sit in Zanin for the night. So some of them stay in that area. Some of them fly into the cities. Some of them, this dot here is a mine kilometers away. And the bat just goes and sits there, and there's no other bats there, so I'm not sure. But all of them have a different activity that they do every night, but most of them stay in that valley or just go over the mountain to Zanin. So this is giving us some data. Why is this important? So I want to link this to some of the work that Jonathan Towner and Brian Amand did at CDC recently, where they spiked fruit with Marburg virus in the lab. And they tested how long the virus will be viable. Because we had some Marburg outbreaks that was not connected to caves. And we're trying to figure out what is the interface. Can it be fruit that's got the virus on that somebody picks up or an animal picks up? And what they found with this is that Marburg virus specifically can survive on fruit for 6 hours. And they did virus isolation. So this is live virus. It's not just nucleic acids. And that links to some of the movement data that we're looking at. So if a bat goes and it hangs in somebody's fruit tree till 5 o'clock in the morning, eat on a fruit, and that's what they do, and spit it, half of it out, lands on the ground, pig comes or goat comes, they eat it, or somebody picks it up because the fruit only have a little bite maybe in it, that risk interface is there. So it's really something that we need to look at in terms of looking at risk. And then some of the last things that I'm just quickly going to show is how does human activity influence all of this? And this was also work done by one of my previous postdocs, where we just looked at land changes around important caves, and this is important bat roosts in South Africa. You see all the dots here, maternity roosts and some of the other major roosts. Just used existing land use data, so it was done very high level. And then we also looked at how close people are moving to these caves. And we did it for a period from 2014 to 2018. And just in those 5 years, we had a 4 to 10% change in woody vegetation. So that means trees are removed around these roosts. And that's a high percentage for that short time period. And we could clearly see how houses are moving closer to caves. So this brought up the discussion about buffer zones around caves and conservation and having policies to protect caves in South Africa so that we don't have this close contact with bats and people. So what's next? So we just started with a human behavior study in this area, which is linked to questionnaires. So we're going to ask questions about bats, but we're also going to ask questions about other wildlife interaction and livestock to try and see what may be the interfaces in South Africa. And this is important because we can't assume that people are eating bats in South Africa and that's going to be a risk because people are not eating bats. That's an issue in some of the other countries. Our risk interfaces may be something completely different. Maybe people going into caves for religious reasons. And you need to understand in the context what those specific risk interfaces can be. And this brings me to the final part on serology. So serology for especially bat pathogens and most wildlife is really complicated. It's things that we need to develop. We're dealing with a high diversity, so you can't just use known viruses because you get cross-reactivity. You don't know what the cross-reactivity are. And with bats, you get very small amounts of serum. It's okay if you do human surveillance or if you do livestock. But for insectivorous bats, you mostly sit with 10 microliters. And then you have to decide what assay you're going to do, and there goes your serum. You can't even do anything follow-up. So you really need assays that can accommodate very low amounts of serum for a large diversity of pathogens. And we decided to move into the Luminex Bioplex systems, where you can then develop antigens that has to also be representative of the wild type. So this is not so easy. This is all to do with expression and getting functional antigens and having the sequence of the proteins first, and then coupling this to different beads where you can read different fluorescence. And we do this with Uniformed Services University, and we've developed now good assays for filoviruses, full filovirus diversity, not just Ebola and Marburg, paramyxoviruses, and we've also now just done it for coronaviruses. And then we include things like HKU9 if we have a specific virus that we want to track in a bat population. So it's very easy to do. You throw your things on the machine and you get a result. But where the tricky things come in, how do you analyze this cross-reactivity? And this becomes, again, very fancy stats, very fancy modeling to try and decipher the cross-reactivity that you're getting. But it can give you an idea of if spillover happens. If we test livestock, we can see there's some signal. If we test humans, and we're going to collect serum now with our human behavior study, there's signals that's coming up. So we've done this for bats, and this is, I think, almost the last slide, where we did it for filoviruses, and we know we've got Marburg in our fruit bats, so we get seropositivity for Marburg. But we're finding another virus that's between one of the new filoviruses, Bombali, and Ebola. So we know there's something there that's not Marburg, it's not Ebola, and we can now try and target nucleic acid surveillance maybe more towards that diversity, but also try and see what other signals we're getting. And we will then expand on all the bat work in terms of serology. And all of this translates to what we communicate, which is very basic at the moment. It's not on policy level or on recommendation level yet, but we try and do community education, and specifically through using books like Living Safely with Bats, translating it into all the South African languages, and trying to make it context specific. This was developed by the Predict international team. So there's some changes to really make it context specific for South Africa. And all of this comes back to prevention, really trying to address all those 4 red blocks on looking what's going on in host populations, really looking at anthropogenic factors, integrated surveillance, and then risk mitigation strategies. And we won't prevent everything, but if we can at least prevent half of it, it is much more economically better than trying to respond the whole time when people are starting to die, or if we have these outbreaks in human populations. Thank you.